All right. So it's my pleasure to introduce another of our snack seminars. And we're very lucky today to have Anita Luthi, who is coming to us from the University of Lausanne, where she does, I guess, systems neuroscience type work. But Anita has a particular interest in sleep and sleep rhythms and noradrenaline, with having done beautiful work on all of these kind of sleep rhythms and the thalamus. And I think with a kind of particular focus to really understand how these things are linked together, and not just how we're seeing changes in the brain, but also in the periphery. And I was really lucky to meet Anita in person last year at a meeting in Innsbruck, and to even find out that, on the back of all this amazing work she's doing in sleep and noradrenaline, that she's been sort of working on sort of some new hypotheses that challenge ideas about microarousals. And I really loved that kind of way of thinking, because, taking this thing that everybody thinks is a problem, but actually asking, hang on, why does the brain do this, instead of just thinking it as a problem. So I think we're very lucky to have Anita talk about her work and some of the kind of bigger ideas about sleep and noradrenaline. So welcome. Thank you so much. And yeah, when you're ready, you can share your slides.
Okay, so again, good morning to everyone. So for me, it's good morning. And yeah, of course, I'm very happy to be invited to this seminar series that I listened to a few and that I really enjoyed a lot. And yeah, thanks to Claire for making this possible and for the very nice encounter we had in Innsbruck and the contacts we've been having since then. So let me share my screen. So can everybody see this?
Yeah, that looks great.
Okay, and I'll go with my laser pointer.
Great. We can see that too.
Good. I
Thanks.
minimize that one. Okay, so yeah, I was going to talk to you today about mostly sleep and the neuroscience of sleep from a particular point of view, which is that we often see or typically describe sleep as a disconnected state. But at the same time, I think it's also clear that we should remain vigilant during sleep. So we are not totally disconnected. And that sort of leads to a dilemma of how these two opposing sleep functions can be merged and reconciled together. So that was sort of the overarching question we've been having for a number of years now, and we brought it forward to also find underlying neuronal mechanisms. So this is mostly an animal experimental talk. I'll allude a bit to some human sleep-related work that we are also doing. Feel free to interrupt or ask questions anytime if I'm going too far in experimental details or you want to know more about the experiments. I really see it as a conversational talk, and I'm fine with being interrupted anytime. So to get started, so falling asleep, one characteristic of it is that we are getting disconnected. And much of how we describe sleep today is about getting disconnected from the sensor environment. So what you see here is portions of hypnogram of a human person who is recorded with EEG, and based on these signals, you can subdivide his vigilant states from wake into different stages of non-REM sleep, and 1 and 2 and 3, and also REM sleep. And all these stages have characteristic EEG, EMG signatures that I will explain in a while. What is best known, I think, to us is that we think of certain non-REM sleep stages as light sleep stages. These are the ones that we enter right from wake sleep. So you have N1 and N2 sleep that are characterized as light sleep. And light sleep means that these are sleep stages where it's rather easy to wake up from and where the arousal threshold is low. In contrast, we speak about deep sleep. That's the N3 sleep state. That is the sleep state where it's harder to wake us up and where the arousal threshold is high. And this can continue to REM sleep. I will not talk about it right now because it's a state that I'm not going to enter much into what it means to be disconnected there. So much of what has been thought about, what are the mechanisms that underlie these different degrees of sensory disconnection has been influenced by the so-called thalamic gating hypothesis that puts the thalamus as a center stage, which gatekeeps information flow from the sensory environment, and the particular state in which the thalamus is in would then determine the extent of information flow or sensory information flow into the cortex. And for this, for example, the sleep spindles that are shown here are important. They are generated in the thalamus, and it's thought that these are important in regulating that information flow. Beyond the thalamus, also slow waves are important. They are generated in the thalamocortical circuits, and the extent and presence of these slow waves also plays a role in sensory information flow. So falling asleep is largely getting disconnected, but at the same time, we also know that falling asleep also means that to some extent we remain connected and we remain vigilant. So for example, we can discriminate our own name, or a mother can discriminate a baby's cry from other noise, and we would preferentially wake up to our own noise. During sleep, we have some capacity of semantic and numerical elaboration. And it's also clear that during sleep, we continue monitoring essential life functions, in particular in relation to bodily activities. Electrophysiologically, it has been shown that during sleep, we preserve sensory evoked responses also in primary cortices. So this challenges the idea of the thalamus as an absolute gate. We also can generate motor preparatory responses during sleep, and sleep is full of spontaneous arousals, which are sort of stereotyped thalamic activation, also cortical deactivation processes that have been multiply described in human sleep. So sleep is not a steady state of disconnection. It's also not a state of steady disconnection, but it suggests that there is some kind of fluctuation that must be ongoing to link the state of disconnection with that of the remaining connection. And that idea of arousal fluctuations is not a new one. It's indeed something that we think of very naturally if we think of wakefulness. For example, behaviorally, we know that wakefulness can be divided into more quiet states of wakefulness or more attentive states of wakefulness. Physiologically, we know that if we go into states of attentive wakefulness, there is parameters both at the central but also at the peripheral level that change in a concerted manner to allow this increased levels of arousal that necessitate to go into attentive wakefulness. Finally, also, even if we are disengaged, if we are in resting states, there is evidence for resting state fluctuations of functional connectivity measured in bold imaging, where certain brain areas that become disengaged during tasks, that's shown here in these cold colors, remain active and continue fluctuating in a concerted manner on very slow timescales. So really what we were interested in is finding out what are arousal fluctuations in sleep, what are their dynamics, what are their mechanisms, and how can we understand them in terms of the larger concepts of getting disconnected, but also remaining vigilant. And we want to study these arousal fluctuations in mouse. So we are an animal experimental lab. We do sleep studies in a routine manner. And the question was, how can we disconnect and remain vigilant? How would that work together? With the ultimate goal to identify neuronal mechanism that would support such fluctuations and also making our mouse work relevant for human sleep work. So this is how it got started a few years ago. So here you see how we implant our mice with EEG, EMG electrodes, and typically also a number of LFP electrodes that allow us to record local sleep signals, and they will become important at some point. Our animals are then allowed to move freely and sleep freely in their cage. They are tethered, and this produces sleep-wake behaviors that we call undisturbed sleep-wake behaviors. So here you see a typical hypnogram of a mouse in its cage at the beginning of the light phase. That's the predominant resting phase of animals. And the animals then stays awake at the beginning, then falls asleep. There is only a single non-REM sleep state in mice that is scored. It stays in there for repeated bouts. It's interrupted by wakefulness and also occasionally goes into REM sleep. So a very polyphasic sleep-wake behavior that is really quite different from the one that we have in human sleep. But nevertheless, there are consolidated bouts of non-REM sleep here that I highlight in green and that we took in the first place. So focusing on some limited period in the light phase, we took these bouts and we just asked, what are the dynamics of the sleep-related patterns in these bouts? Are there any inhomogeneities that could be of interest for us? So we initially did that in a very simple way using 4-second epochs and doing spectral analyses on these and then plotting band-limited power dynamics across the successive power spectra that we extracted from the 4-second epochs. So here you see this for two characteristic frequency bands. On the one hand, you have the sleep spindle that we also call sigma band. That's the 10 to 15 hertz band shown here in red. And we have the slow wave activity band that is the low frequencies that include slow oscillations, so 0.5 up to 4 hertz. That is shown here in blue. And you see these are high, there is high power in these frequencies because that's typical for non-REM sleep. So when we did that and looked then at some natural behaviors, sleep-wake behaviors of these animals, we quickly realized that there were some interesting dynamics that had not been described before. So here you see about an hour of sleep-wake behavior in the early light phase of the animals. And now plotted here the sleep spindle band, 10 to 15 hertz slow wave activity band, as I just said. Done here specifically for all the non-REM sleep bouts, and then we stop them once we go into REM sleep or wakefulness. And what should jump to eye here is that in particular, the sigma band shows these very vigorous fluctuation around the mean power levels that are shown here. That show a kind of oscillatory behavior that are in particular pronounced when you have long non-REM bouts. So here several tens or several minutes, you have pronounced fluctuations in the sigma band, but much less clearer or more variable activity in the slow wave activity. It's just constant and high, which is typical for non-REM sleep. Are you all still around? Is it all clear? I'm slowly getting started here. Okay,
Oh, it
you
seems
tell
clear.
me if
It's
everything
good.
is not
It's
clear.
good. Thank you.
Just begin now. Okay, so when we then analyze these patterns and do power spectral analysis on these frequency bands, what comes to eye is that the sigma band shows pronounced low frequency components that go down to very low levels and have a peak of 0.02 hertz. So that's about 50 seconds here. So here individual animals are shown in the green line, in the gray lines, and the red line is the mean of, I think it's a good dozen of, it's 10 animals here that are meaned. So these 50 seconds are really what you see in this plot here. So that's sort of the interval between these peaks. And you see this is a very broad peak. So as soon as you move a bit off, you go into another frequency, so a much faster one or a much slower one. But really the peak around 50 hertz was, 50 seconds was really very consistent. And it was not present as clearly in the slow wave activity. So it's for a similar analysis here, and if we did this across a number of bands, so slow wave activity, theta, then sigma and beta, the sigma really came out as the one where these inhomogeneity, so these fluctuations, were the strongest. So this was interesting because, as I mentioned at the beginning, sleep spindles are related to sensory disconnection. So such a pattern would suggest that animals fluctuate between more or less sensory disconnection. And it became even more interesting when we also looked at other parameters that we thought could be interesting for arousal-related fluctuations, and these involved the autonomic nervous system. So at the time, we did not have much access to autonomic parameters, so we simply took our EMG, which is a nuchal EMG, but if it's well positioned, you can very well pick up the R interval, so the heart rate. And then when we detected the peak and plotted the heart rate, we could also see variations in heart rate. And when we plotted such variations, so here you have the heart rate in red, and in blue you have the sigma power that I just showed you before. It became quite clear that the heart rate tended to shoot higher when sigma power was low, and it tended to go down when sigma power was high. And this pattern continued all over. So you see, it's variable, it's very much noisy, but when we constructed anti-correlations on individual bouts, it was very clear that there was a strong anti-correlation, meaning that when sigma goes high, heart rate goes low. So a very peculiar, unexpected correlation. I can tell you how we got about looking at heart rate later, but at the time it was very reassuring for us to find a similar pattern of activity in a variable that seemed to be independent. And so the question was, of course, is these fluctuations really relevant for arousability changes? So what we then did in our mice is we recorded them again for sleep-wake behavior. And then at the beginning of the light phase, so in the first hour, we played sounds to them. So simple white noise in their cage. And we chose that intensity such that the animals would sometimes wake up through the noise and sometimes they would sleep through in response to the noise. And this was measured by simple EEG, EMG. So the wake EMG signal would show an activity, and the EEG would desynchronize, whereas when the animal would sleep through, we would not see such changes. And then we looked basically when the animal woke up or when it slept through, at which phase of that infraslow 50-second fluctuation we had observed previously, at which phase that animal had been. And what we saw is that as soon as the animal would wake up, the noise that we had played randomly, so we did not know at which phase the infraslow fluctuation was, when the animal woke up, the sound had started at the moment that sigma was high and started decaying. And the converse was true when the animal slept through. So at that moment, sigma had been low and started increasing. And when we plotted all these sigma power dynamics as a function of what happened to the animal at point zero when the animal was exposed to the noise, we got evidence that there was an opposite relationship for wake-ups compared to sleep-throughs. So the wake-ups would take place when sigma started to decay, and the sleep-throughs would take place when sigma power started to rise. So that was all not very highly time resolved and not very easy to do, also because waking up animals is a tedious process. You have to wait until they fall asleep again, and so the data set was not very high. But if we put it all together, we arrived at the following scenario, namely that we have a very slow variation in sleep spindle activity in freely sleeping mice during non-REM sleep. We later detected these spindles, you will also see that. When animals are in the phase of high spindles, they tend to sleep through, whereas when they are in a phase of low spindles, they tend to wake up, or in the phase of decaying sleep spindles. All this happens on top of consolidated non-REM sleep. The heart rate shows an opposing behavior. So when the animals are in a more protected mode, heart rate is low. But when the animals anticipate a potential awakening or wake up more easily, heart rate would go up. So we have constellations of more disconnection and less disconnection, so more vigilance that alternate on the 50 seconds. We were not the only ones looking at this, but around the same time as us, a colleague from us, Daniel Huber in Geneva, found that fluctuations in pupil diameter also coincided on this time scale and were also related to visually evoked awakenings in mice. So that reassured us further that we had some pattern of activity that seemed to be behaviorally relevant. Now, this 50-second time scale also is not a new one. It has been described already in the 50s by a Russian who studied rabbits and who had done EEG recordings and seen that there were slow electrical processes in the brain with slow fluctuations at the cortical level. And she had speculated at the time that this could lead to variable cortical reactivity. Also, the 50-second is not a new signal, but it's known for fluctuating arousal levels in bold imaging studies. So the very first ones looking at fluctuations in resting state activity had come up with this time scale. Here, this is a newer study from Raut et al., who has even more elaborated on the spatial temporal dynamics of these fluctuations. But also in old sleep studies, where people had been looking at a pattern that is called cyclic alternating pattern, where you have alternating low frequency and high frequency activities on a 40-second time scale. This had been related to fluctuating arousal level, although it remains a bit hard to study until today, and it's not applied by everyone. But also at the level of other signatures such as K complexes that I will get to later, slow recurring patterns have been described. And last but not least, from my very early work as a postdoc in the McCormick lab, I had studied thalamic circuits activity in vitro in the slice, and I had looked at sleep spindles and their refractory period that separates individual sleep spindles, and these were also taking place on tens of timescales. So I had been sort of sensibilized to this timescale already thanks to my postdoctoral work, and I was convinced we should go a step further and try to find out mechanisms. So this is really the very initial sort of observational work that we had done that I bring forward here, that we have confirmed later with more timed interventions. But for today, let me now go into the neuronal mechanisms. So we started looking at this based on sort of an educated guess, also based on the work I had done in the McCormick lab. We had been interested in the LC, so the locus coeruleus, a brainstem area that is important for noradrenaline. I don't think I need to introduce that in much detail here, but this is an area that is very interesting because it coordinates arousals in the central nervous system and also in the periphery, in the sense that it can coordinate increases in high frequency activity in the central nervous system with changes in autonomic parameters due to its its forebrain and also its projections and also its brainstem projections. Furthermore, so there is many people who have worked on this, and furthermore, the LC has really been involved in arousal very tightly from very early work done in the Aston Jones lab, working with monkeys, recording LC activity as the monkeys were falling or alternating between wakefulness and sleep. And also more recent work in rodents where optogenetics can be used to target LC neurons that are shown here in green that express an optogenetic actuator. And if you then turn the laser on that is connected to an opaque fiber in these animals, you can, by simply stimulating the LC, you can just induce a full-fledged sleep-wake transition. So we thought, let's look at LC activity a bit in more detail during sleep. But before going into all the optogenetics, we first started with some pharmacology experiments. So we used our mice to not only do sleep recordings, but also we introduced a craniotomy that would allow us to introduce pharmacological agents over the thalamus to interfere with noradrenergic activity. And we chose the thalamus because we knew this is an area where spindles are generated. So if these spindles are relevant and the LC plays a role in the regulation, we should find changes in spindle activities there.
Anita,
And
do you know,
yes.
are you able to say kind of where in the thalamus you're targeting, or if this is if it's the pharmacological manipulation, then I
Yeah, so no, I should be more specific. So we had a lot of experience by looking at S1, so that's somatosensory
Hello, hello.
cortex. Um,
Ah,
hello,
shoes.
uh,
that somatosensory cortex where spindles
sorry,
can be
Oh,
recorded.
idiot.
Jordan,
Oh, it's because your feet are so
can you
big.
mute yourself, please?
Oh,
Th-
sorry. I didn't realize that I'm moving.
Thank you. Sorry.
No problem. So it's primary somatosensory cortex it's in the whisker-related area, and so we introduced that pharmacology in the somatosensory thalamus, so ventrobasal complex. It's not very specific because I mean you infuse that drug, but that's the area that we had linked to sleep spindle generation in the primary somatosensory cortex in earlier studies. So we knew that if we infuse drugs there, we could record sleep spindles in S1 using an LFP electrode, and that would report to us on the corresponding thalamic activity. Yeah, sorry for going a bit fast.
No, no.
So in any case, so we recorded these animals in freely moving recordings, and we habituated them to being head restrained so that we could inject drugs for a few days. And then we always recorded sleep. And then there was an experimental day where we infused either drugs or ACSF in two separate groups of animals. And so here is now the result. So you have here a baseline animal recorded with its hypnograms. You have wake, non-REM, and REM sleep again. And here you have now individual spindles that we detect and the corresponding sigma power fluctuation. So you see the tight alignment between the sigma power and the spindles that come in clusters around this time of the sigma power. So these are spindles taking place at the same time. They're just vertically shifted so you can see the individual ones. And when we now, in that same mouse, a day later, we injected atenolol and prazosin. So these are alpha and beta adrenergic receptors, antagonists. What we observed is that the fluctuations in sigma power at S1, cortical local field potential level, they were essentially gone. Spindle activity was way above average and much more constant, as you can see here in the individual spindles. In contrast, when we did animals in which we just injected ACSF, so artificial cerebral spinal fluid, so it's essential NaCl, we could not reduce these spindle activities in such a dramatic manner. So we analyzed the strength of the fluctuation by this power analysis that I showed you before, and it was clear that this fluctuation had disappeared, whereas spindle density had gone up. And in the control animals, we had some diluting effects due to the ACSF injection, but overall, the spindle pattern was much better preserved. And so this encouraged us now to go and do the whole battery of animal experimentation work that is possible nowadays to really focus on LC neurons and to manipulate them using targeted methods. And so for this, we use mice that are a Cre driver line that is driven by dopamine beta hydroxylase. So that means the dopamine beta hydroxylase, that's the enzyme that is responsible for noradrenaline synthesis out of dopamine. And in these mice, the promoter of this gene is linked to a Cre recombinase, and the Cre recombinase allows for selective expression of genes of interest, such as, for example, channelrhodopsin. These are very specific targeting methods because the large majority of tyrosine hydroxylase positive neurons, which is another marker for noradrenaline, then, for example, can be led to express mCherry, which is just a fluorescent reporter. So you see here the nice overlap between the green TH neurons, so these are all the noradrenergic neurons, and the red ones, which are the transfected neurons. By viral transfection, you can introduce that mCherry, and then you have the merge, which is the yellow color, and most of the neurons express both colors. So with these mice, we can combine sleep recordings, EEG, EMG, local field potential recordings. We can introduce optic fibers, and then we can either use sensors that allow us to monitor the activity of LC neurons. So these are fluorescent molecules like GCAMP that become fluorescent once LC neurons are active, once they have a calcium increase. So we can directly monitor LC activity while the animal is sleeping. We can also monitor free noradrenaline levels nowadays because there are specific biosensors for free noradrenaline. That's the GRAB sensors that we can also introduce virally. For example, here we do that in the thalamus, whereas the biosensor for GCAMP would be introduced into LC. On the other hand, we can do optogenetic interference. So we can introduce channelrhodopsin into LC and manipulate, stimulate its activity. Or we can do the converse, which is optogenetically inhibit LC, so by introducing JAWS, which is an inhibitory opsin, so we can block its activity. So there's tons of experiments that we did in this direction, and in particular, what we did also is to do this in closed loop feedback. So using the sleep signal, including also the spindle signals, to drive what we want to do with LC. So, for example, we could monitor sleep spindles and then stimulate LC and see what happens on the LFPs. So one of the very most simplest experiments that we did is just the bidirectional optogenetic manipulation of sleep spindle fluctuations by stimulating or inhibiting the LC using optogenetics. So we either injected channelrhodopsin into the LC that would allow us to stimulate it, or JAWS to inhibit it. We combine it with sleep recordings and sleep electrode implantations, and then we let the animals freely sleep. And as soon as the animal went into non-REM sleep, we would turn on blue light, either to stimulate channelrhodopsin, or orange light to activate JAWS, and that would inhibit the LC. And here you see the corresponding histology that shows you that we can specifically manipulate LC. And the response was very clear. So here you have an animal that sleeps freely, and that alternates between sleep and wakefulness. It has infraslow sigma fluctuations, it has sleep spindles, and at this moment here, we turn on the blue light, so this animal expresses channelrhodopsin, and immediately sleep spindles have completely disappeared, but the animal keeps sleeping. So this is a very fine manipulation, a very moderate stimulation of the LC to not wake up the animal. So it keeps sleeping, but it sleeps without spindles. And as soon as we stop the light, the fluctuations come back. You can see that the strength of the 0.02 fluctuation goes down, and the sleep spindle density goes also down. So LC activity induces a state of low spindle, so a state of higher vigilance, of greater connectedness, if you want to link that to what I showed you before. Conversely, when we do the same experiment, but we inhibit the LC, what you can see is that the amount of sleep spindle increases, they get more dense, and the infraslow fluctuations, they become sort of stuck at higher levels. They no longer decay as easily as before. And when you quantify this, you obtain a greater density of sleep spindles and also a blockade of the 0.02 Hertz fluctuation, although that is more moderate because inhibition, I tell you, experimentally is a more difficult thing to achieve. But in any way, what we could show with this is that the fluctuations are very, very sensitive to LC activity, and even moderate stimulation, so a 1 Hertz stimulation of the LC can completely erase a sleep spindle activity. So that's one experiment we did. Another one, just to move on a bit more quickly, you can target your optic fiber either to the thalamus, so the ventrobasal complex here, or to the cortex. And you can then ask, where does stimulation of LC fiber affect spindle activity in the cortex? So we could either stimulate the fibers in the thalamus or in the cortex and then monitor what happens at the level of sleep spindles. Here is the corresponding anatomy showing that axonal fibers are present at both levels, both in thalamus and in cortex. But what we found is when we stimulated LC fibers in the thalamus, in VPM, we could suppress spindle activity as we had done before when we stimulated LC directly. Or you can, if you stimulate LC fibers in S1, we did not get such a strong effect. So the oscillations were largely preserved. So that confirmed that LC fibers, so axonal projections into the VPM, were the ones that were largely responsible for sleep spindle regulation at the cortical level. Is that clearer?
Yeah, no,
So,
that's
so the LC projecting, making wide projections into the forebrain, both at the thalamic and into the somatosensory cortex, and we record these spindles locally using our LFP electrodes in S1, and you can clearly see that these are thalamically driven fluctuations that depend on LC activity.
Anita, do you have any ideas of, because I guess in my, simplistic understanding of sleep spindles and their generation in the thalamus, I guess I always thought about the TRN as being the kind of initiator, I guess, of the spindles. And then, the interaction with TRN and thalamocortical projections kind of sustains them. Do you think in the context of kind of what you're showing here and the LC projections, do you kind of have an idea of what the TRN is doing?
So we have recorded from both VPM neurons and adjacent TRN neurons in vitro.
Yeah.
So in slice preparations using patch clamp, and then we optogenetically stimulated LC fibers in the slice, and both types of neurons depolarize very rapidly in response to that stimulation. So both thalamic and TRN neurons are sort of brought into a membrane potential regime where they no longer generate sleep spindles.
Okay.
So it's by in the VPM, it's a beta adrenergic mediated depolarization that is induced, whereas in the TRN it's more alpha 1 receptor induced.
Okay.
So this is all shown in this paper that we published a few years ago.
Oh, cool.
So I think this was probably the most complicated part when it comes to animal experimentation.
Yeah.
And now it's getting a bit simpler because I'm going into now, not showing all the optogenetic experiments that we did originally, but really shifting to this very direct measure, which is just monitoring LC activity directly using fluorescent biosensors for calcium activity. So we have here GCAMP8S, which is a molecule that becomes fluorescent once it's encountering calcium. And this molecule, we express it in LC neurons using viral targeting strategies, and then we monitor the corresponding signal. And we also use another sensor, which is a sensor for free noradrenaline, the GRAB-NE1 that I mentioned before, that monitors on released noradrenaline that we can also express virally. So we literally can in the mouse monitor both LC activity and the corresponding release of noradrenaline at any site that we are interested in the brain by just locally expressing these sensors where we want them and then introducing optic fibers. So this is really amazing because literally millimeters away from the site of origin, you can see what happens. And of course, we were interested in the thalamus as the site for sleep spindles. So this is how this would look like. You have here a mouse, a DBH3 mouse that is injected with this biosensor, and you can see these neurons then express this. This is a green sensor, and you see how it co-localizes with TH, which is a marker for noradrenergic neurons. And if you overlay, you can see that the substantial fractions of TH expressing neurons also express this biosensor. And here you can see the optic fiber that is positioned on top of the LC. So we can really pick up the fluorescent signal that is generated by the GCamp. And so this is one of the first signals that we got by doing such an experiment now in a mouse that we record for sleep-wake behavior together with the corresponding LC activity. And so this is now sort of the direct measure of LC activity that we had sort of read out in our initially naive behavioral assessments of arousal fluctuations using sleep spindles. And the result was really striking because what you can see here is that LC activity overall, measured at the population level, stays high during wakefulness. This is typical for LC. But as soon as the animal falls asleep, the LC activity decays to a lower baseline, on top of which you have very marked fluctuations. So the LC is far from silent, and it does not fall silent until the animals enter REM sleep. And what is remarkable is not only this recurrent activity, but also the extent of it. So the population activity becomes as high as in wakefulness, yet the animal from an EEG point of view is asleep. If we plot this, taking mean values, so we take all these points and we mean them per animal, we get wakefulness having high LC activities. We get non-REM sleep having intermediate activities. That's no surprise because we have a fluctuation between high and low levels. And we have REM sleep having the lowest levels. And that's how usually LC activity has been seen, being high in wakefulness and then low during non-REM sleep and lowest during REM sleep. But if you plot this data in an all points histogram, which is shown here, it should be clear that this graph does not do justice to the real situation because non-REM sleep is really fluctuating between high and low levels that cover the ones of REM sleep and that cover the ones of wakefulness. So it's going back and forth between these. So here we have really just plotted all the points for a single animal, of course, because these signals depend on the dynamic range. Okay, so very vigorous LC fluctuations. If we now combine these measures, so we measure LC calcium activity and we measure noradrenaline release in the thalamus together with S1 sigma power, so the infraslow fluctuations, not super beautiful here, but then the CA1 theta delta ratio, so that's for scoring sleep, EMG. What jumps to eye is that you have both LC fluctuations and you have thalamus noradrenaline fluctuations. And if you zoom into these, you see that the LC activities fluctuate and the noradrenaline co-fluctuates with it. So these fluctuations during non-REM sleep, they have a significant output in the thalamus. You have very marked alterations in noradrenaline levels, and that explains why spindles fluctuate. So altogether, what the state that we are at for the moment is that LC is a structure that is highly active in non-rapid non-REM sleep, non-rapid eye movement sleep. It's shown here schematically. You have fluctuations on the 50-second level. They are coordinated with sleep spindle activity in the brain and heart rate, and they are causally involved in this. So this we have shown optogenetically. I've given you one example for it. This coincide with arousal fluctuations. So mice tend to wake up when LC activity is high, and heart rate is high, but spindle power is low. And low arousability when the converse is true. So when sleep spindle activity is high or starts decaying and LC activity is low. So this is also interesting because it means that LC activity divides non-REM sleep into substates that are characterized by either high, by a surge of LC activity, or by a trough. So the LC activity surge is the one that controls arousability. That's the one where arousability is high. And the troughs are moments where LC activity is involved in other aspects, in particular in relation to REM sleep control. So LC activity has to be low for REM sleep to be entered. So you really have a subdivision into these two states. And for the rest of this talk, I will mostly talk about the surges because they're related to arousals. And in this next figure, I show you that not all LC activity surges are in fact equal. So you have, if you look at the hypnogram, you have wakefulness and non-REM sleep, and you have also brief awakenings that are called microarousals. And if you look at the corresponding LC activity, you see that LC activity is high when the animal is awake. And this is also when thalamus, LC, and noradrenaline levels are sort of steady. And then you have the fluctuations in LC activity during non-REM sleep. And if you look at these, so first of all, you have high LC activity levels during wakefulness, behavioral wakefulness. Then you have the ones, the LC activity surges that coincide with behavioral arousals during wakefulness. So these brief arousals that we also call microarousals, and I'll show them to you in a second. So then LC activity is high and corresponding noradrenaline is also high. So these are the microarousals. But then at the same time, you have arousals during which there is no signal. You have LC surges during which there is no arousals that are seen at the polysomnographic level. So this is, for example, this one here, where you see LC activity is almost as high as during a microarousal, but and noradrenaline is also high, but electroencephalographically we cannot see a wake up. So this suggests there is different levels of arousal, different states of arousal that depend on the exact extent of LC activity. So the question is, what are these arousal states? What arousal levels are they? Are they local versus global? And what are the polysomnographic readouts of an LC, more arousal surge-related activity, but you cannot see it in traditional measures of arousal? So in this plot here, I show you now a more in-depth analysis of non-REM sleep spectral dynamics. You have the corresponding LC signal here, the different surges. You have the sleep spindles that are, you see they are high when LC activity is low. You have the LFP signal in the sleep spindle band here that is also high when spindles are high. And you have also the microarousals now indicated throughout all the signatures. And what should jump to eye here is that every time there is a microarousal, LC activity is also high. So microarousals coincide with high levels of LC activity. And microarousals are really arousals in the sense that you have a desynchronization of the EEG and you have an EMG activity. You have one here and you have one here. This is a stronger one, this is a weaker one. So microarousals, so these brief interruptions of non-REM sleep are the ones that they coincide with LC activity. And this is also shown here in the plot where we plotted thousands of microarousals from tens of mice all together as a function of the phase of sigma power. You see all microarousals fall into the trough of sigma power, which corresponds to high LC activities. So LC fluctuations guide or coincide with these arousal fluctuations at the microarousal level. Now, if we plot LC activity peaks, so the surges that coincide with a microarousal. These are the ones that are seen here. So you have LC and LC surge. You have a decrease in delta activity, so low frequencies go down. That's typical for arousal. We lose the sigma power. That's also normal because we have LC activity high, and we have an increase in gamma. So these are standard signatures of arousal that typify such a microarousal. In contrast, if we take the LC activity searches for which we see no microarousal, we have an increase in delta activity instead, so we have not a decrease in low frequency activities. Sigma goes down also because that's the sensitive marker for LC activity, but also gamma power does not go up. So this is sort of a non-standard type of arousal where neurobiologically we have higher LC activity levels, but polysomnographically it does not look like an arousal. So you really have two types of arousals that are different from an electrophysiological point of view, and that's all quantified here, where I hope you can clearly see how LC activity surges vary as a function of amplitude, depending on whether you have an arousal, you don't have a microarousal or a short or a longer one. And this is the delta power that you see is going up, is positive at the level of no microarousal, but is suppressed once there is arousal. And this continues like this for the other frequency bands. So higher LC activity once you go into a microarousal, decrease in delta activity once you go into microarousal, but higher delta activity when there is no one, and then the corresponding changes for the other frequency bands. So are all LC activity searches then really an arousal? Because the EEG signature doesn't really show that. No, it looks more like you have an increase in low frequencies that are more typical for sleep. So we looked at heart rate variations as a function of LC activity searches when they were paired with a microarousal or without a microarousal. You see LC activity searches are smaller in that case than here, but nevertheless they remain associated with a small increase in heart rate, whereas it is large for a microarousal, which is a full, more wake process. But still, you have a small autonomic arousal that remains present, even if in the forebrain you don't have an arousal. And that is shown here with the very tiny increase in heart rate for no microarousal and the full-fledged one once you have a microarousal. So this shows that LC does generate arousals, but they vary in terms of the extent of the autonomic and the cortical levels of arousal. So that's why we came up with this proposition that there are two different types of arousal that coincide with graded levels of LC activity in mouse. So here you would have a subcortical autonomic arousal that comes when LC peak activity is sort of moderate, that suppresses sleep spindles, but in the cortex the activity is not strong enough to really induce a suppression of low frequency activity. Instead, you have an increase in that low frequency activity and a moderate increase in heart rate. In contrast, for a microarousal, so for a full cortical autonomic arousal, you have higher LC peak activity, sleep spindles are also going down, and you have a disappearance of low frequency activity because the activation of the cortex is stronger and also autonomic arousal is stronger. So this is a very interesting observation that is currently really followed up also in other studies. So you have a new paper that just came out from the CCLARI lab, where they have done a comparison between the spectral changes that are taking place during an arousal in humans, where you have an increase in high frequency activities as the human transitions from sleep to wakefulness. But what they observed is that under these conditions also there is always first an increase in low frequency activity before the higher frequency occurs. And this also makes sense in relation to fMRI studies that were done recently in the Laura Lewis lab. And it fits very well with what we have observed in our animal research. And the interpretation of what that they bring forward is that there is sort of a two-stage activation process that is taking place from the LC that involves activation of thalamic nuclei, and that can generate low frequency activities if this activation is moderate, whereas a higher frequency and a higher activation of the LC is then required for more desynchronization at the cortical levels. And they propose that in fact K complexes, which are these very unusual signatures that one sees in human sleep, but not in rodent sleep so far, that are typical for N sleep and that involve an increase in low frequency activities, that these could actually be a reflection of LC activity in human sleep. So I think I'm pretty much done with my time. I don't know how should I continue a bit with the human part?
Yeah, I mean, I kind of scheduled an hour and a half, so I guess most people who are keen and want to ask questions, I think
So,
we'll be happy to stay on and hear the human stuff. So please,
okay.
yeah,
Okay, so I continue.
thank you.
So we have also in collaboration, we have also tried to move on with this understanding of infraslow arousal fluctuations, and we have collaborated with Caroline Lussenberger, who is a colleague of mine in Switzerland, and we exchanged a lot. And she had developed in the lab a possibility to monitor pupil diameter in sleeping humans. These people wear goggles that allow them to keep one eye open during sleep. So the eye is stitched open with scotch. But this is in a humidified chamber that is closed, and that so it allows some sleep, actually also deep sleep in these subjects. And in this way, she could monitor pupil diameter and then quantify it and plot it as a function of vigilance state. And this was, of course, interesting for us because we know that pupil diameter is regulated by monoaminergic systems, including in particular the locus coeruleus. And so she came up with these kinds of plots here. So this is pupil size as a function of sleep state in humans and wake. So you have an overall high pupil size in wakefulness, and then pupil size goes down the deeper you go asleep, and it also stays low in REM sleep. So that's an indication of also like a decrease in LC activity. At least it manifests that there is a decrease in arousal levels. But very similarly to what we found also when we did our LC activity recordings, it became clear that pupil size is not just well reflected if you calculate means, because there is also strong fluctuations in pupil size as a function of sleep state. And she focused mostly on N2 sleep. So this is sort of the light sleep state that is highlighted here, and that shows these marked variations in pupil diameter. And when she zoomed into these, so this is shown here. So you have here pupil diameter. You have time frequency plots for N2 sleep. So you see recurring spindle activity that is also plotted here. And then here, this is a central EEG derivation that allows to see K complexes. So these are individual K complexes. And what she found is that indeed pupil diameter fluctuates also, and it does so on a time scale that is reminiscent also of the 50-second time scale. So you have up and downs that take place on that time scale, and that coincide with spindle fluctuations. So you see spindle activity is high, when pupil diameter is low, and it's the other way around. So that really corresponds also to what we find in mice, although the exact phase relationships they are not exactly the same. And voilà, so this is sort of the evidence that at the human level also you have a 50-second fluctuation that is present in sleep and that has parameters that are very similar to the ones that we also find in humans. So there is not just sleep neurophysiology, there is people who are interested in very different aspects of sleep that go beyond spindle measures, autonomic measures, but there is also blood volume changes, there is CSF flow, and this is a figure that I put together with Maaike Nedergaard, who has been very leading this field of CSF flow in the context also of the glymphatic system. And you might have seen one of her recent papers. I should actually cite them here. So this is the paper from Nathalie Hauglund that was recently published. And what they say is that they think that LC activity fluctuations during non-REM sleep, they are important for noradrenaline, they are important for spindles, they are important for the heart rate, but beyond that, they have an effect on blood volume. And this, they say, is likely due to the fact that LC also has vasoconstrictor capabilities. So all these fluctuations, they find in mouse to coincide with blood volume fluctuations that they have also measured using these optic fiber photometric techniques that I showed you. And also they come up even more intriguingly with measures of cerebrospinal fluid flow that is anti-correlated to blood flow, and they attribute this to glymphatic functions. So to the fact that due to vasoconstriction, you enlarge the size of the perivascular space, and that leads to cerebrospinal blood inflow. So the potential functions of these LC activity fluctuations, they are currently, they keep increasing. And the question is really, are these activity fluctuations primarily relevant maybe for these kinds of activity patterns, and these other ones are a consequence of it, or are these multi-parallel functions that are executed and that are all relevant for sleep? And I think probably the second possibility is more likely, also given the fact that there is now new studies coming out showing that LC activity, high and low substages are related to different forms of plasticity and learning capabilities. So we really think we have stepped into something that is very nice to study in mouse. I must say this because mouse shows these fluctuations extremely strongly. They are also found in rats, but when it comes to humans, it seems that maybe they are limited to certain periods, in particular maybe N2 sleep, where the question of being, connected, remaining vigilant and disconnected is maybe still more relevant compared to when you go into deeper sleep stages. But that all still needs to be looked at. So in our lab, we are not going to go into the glymphatic system, but we really would like to focus more on this question of what microarousals are good for and why we actually have them. Because this plot suggests that microarousals are sort of a necessary part or a necessary consequence of all these fluctuating activities. And they might occur in different types of manifestations, sometimes as a full microarousal, and sometimes more as this subcortical autonomic arousal that I showed you. So this raises the question, do different types of arousals have different functions? And how is that balance between regular microarousals and these subcortical forms of arousal really regulated, and how can it go wrong? And with Mike Nedergaard, I'm really sorry because I thought I had added here the citations, but this is from a little perspective that I wrote together with her. We came up with this scenario saying that we think microarousals, as much as they interrupt sleep, as much as they have been seen as actually negative for sleep, so fragmented sleep is not a good thing to have, they are an indispensable part of our sleep because they contribute to its restorative functions in the sense that they generate this blood flow fluctuations, that they generate cerebrospinal fluid, but also in the sense that they are essential for driving sleep between disconnection and between vigilance. And any deviation from this sort of regime in which they are useful is detrimental. So admit that we have some kind of dysfunction of the LC where its activity levels go down. You might imagine LC neurons dying in disease, in neurodegeneration, for example. This is where it's an important topic. You will lose microarousals. You might also lose other forms of arousals that have so far not been really taken into account as arousals, like constellations of K complexes, cyclic alternating pattern related activities. And this has very detrimental effects, of course, on all these glymphatic activities, because this is no longer driven appropriately. It will affect the arousability, the vigilance related aspect of sleep. It will also affect the REM sleep because I didn't show that, but these fluctuations are essential for REM sleep. And it will, of course, affect all the functions that microarousals have been attributed to in relation also to plasticity. So that's one extreme case that still needs to be tested. So that's really a putative scenario that I think with our methods in the mouse, we will be able at some point to interfere with microarousals and test that. On the other hand, if you go into too high LC activity, so we go into states of hyperarousal, we can induce that in mice by just stressing them. We immediately have insomnia features. We have fragmented sleep. We have suppression of REM sleep. So all the aspects related to sleep stability, fragmentations come into play. We might have a disruption of that vasomotor activity also because there is an extensive constriction and not enough moments for relaxation. And also the plasticity aspect, so the importance of having enough spindles for plasticity will also go on. So it's really a collection of phenomena that we try to bring together that I think overall relate to LC activity, but the different types of microarousals could be an accessible manifestation of it in human sleep. So in our lab, if I may just bring up some of the aspects. Of course, we are neuroscientists. We are interested in the circuits, so we are trying to decompose the LC circuit. And here is just some very nice figures collected by Georgios Foustukos, where we have labeled LC neurons based on their projections, where they project to hippocampus, thalamus, prefrontal cortex, amygdala, and we can study their function, their activity patterns in isolation and see how they actually contribute to the phenomena that I've shown you for the moment at the global LC population level. We are interested in global brain states, so we have been pairing up with Valerio Cerbi and colleagues, of which I had the pleasure to realize that actually Max Shine is one of them. And so we put mice in the scanner and we combine scanning with LC fiber photometry. We do that in urethane anesthesia that we found to preserve some of the fluctuations of LC activity. So hopefully we'll be able to show you some new data, and in particular Valerio will share this new data with you. that are very interesting in terms of resting state networks and how they fluctuate. We are very interested in tracking LC dysfunction, as you can imagine. We have projects related to insomnia where we stress mice. You see how much activity patterns change in these mice compared to baseline. We have exacerbated LC activity, fragmented sleep, suppression of REM sleep, and this can be reversed by optogenetic inhibition of LC. We have now a new project related to neurodegeneration together with Matthias Bricke, whom I had the pleasure to know in Innsbruck. So to really see what happens when LC gets dysfunctional, but that's very new and I cannot show any data yet. And of course, we also are interested in multiplexing biosensors. I mean, we should measure more than one signal in one site, so you can combine different colored sensors in the same area. So here, for example, you see LC activity patterns with local noradrenaline release using two-color fiber photometry. That's in collaboration with Tommaso Patriarchi, and the revision of this study is in advanced stage of preparation. So voila, that's a bit the upshot of the whole work that we are doing. I really started with the very old work that brought us to what we are doing today, and it's been a really nice trajectory to discover all this, but this would not have been possible with many amazing people who helped us and provided input. Valerio, Caroline, Yuval, Maiken, of course, that has been a very nice collaboration. Francesca, Ois van Sommeren, and also my local colleagues. And of course, also this work is no longer done by me so much, but really by extremely talented collaborators. So Georgios Foustukos, first of all, he really has introduced the fiber photometry, and he's an expert in the technique. The original work was done by Alejandro, Najma, and Laura, and also new collaborators are now engaged in the new aspects. And also Aurélie Stéphane from Francesca Siclari's lab has now joined my lab, and we are hoping to relate better the studies on what she did in the human sleep on arousals to what we find in animals. So we hope to present some data also very soon. Voila, so I hope that was
Thank you,
accessible.
Anita. That was such a great talk. Thank you so much. It was really amazing to see this kind of story that started with some findings that were probably a bit unexpected, and then how you traced it, you know, for such a long trajectory, but then looking at things across so many different levels to find this whole comprehensive story that is really rich now, but still completely unfinished because you keep finding all these new and beautiful relationships.
Yeah.
It's incredible. It was,
Yeah.
wonderful to see.
Thank you.
I'm hoping that people might have some questions or comments that they had from the talk. But
Feel free. I mean, you could also tell me I did not understand. I will try and re-explain.
No, go ahead. Is that Josh? Did you have?
Yeah. Yeah, thanks, Anita. I really enjoyed the talk a lot. I actually don't know anything really much about sleep, but I kind of like have read things about like how sleep helps with maybe like memory consolidation or behavioral learning. And I was wondering if you have any thoughts about how reducing LC activity in the sleep spindles, but still letting like the animal sleep, if that would have any effect on, behavior or learning afterwards.
Yeah, so that's of course really the burning question, in particular because you have these substates, no, where you have high LC activity, which you would expect, you know, neuromodulatory conditions that are propitious for plasticity, but spindles come when LC activity is low. So it sort of does not match right now very well, but there is new studies coming out now. In particular, there is one from Azahara Oliva that came out earlier this year, where they did pupil diameter measures in mice, and they characterized ripple activity during high and low pupil diameter states. And what they find is that during high LC activity, if I remember that correctly, there is there is a there is a higher density or different properties of ripples compared to low LC activity. And they find that different types of memories are associated with states of high LC activity compared to low ones in terms of replay and also in terms of consolidation. So I really recommend that you have a look at this paper. I think it will all burn down to us not looking at LC activities as globally as we do it still, but really going into, for example, hippocampus, going into the different areas of the hippocampus and looking what the neuromodulatory conditions are at at a given state. So taking the LC activity globally as two different substage, but that means different conditions in terms of serotonin, for example, release in different hippocampal areas, and also, of course, other neuromodulators. And this could mean that different brain areas are in very different states during these two sleep substages that we captured globally for the moment. And I think that this will be critical to understand what happens in terms of hippocampal function during sleep and replay.
Thanks a lot.
Yeah. Maybe in relation to Josh's question, and I guess getting at ideas around the function of these things, which I guess you've, thought a lot about, but it's hard to find the evidence for. If we think about the two, I guess the two types of arousal, so the LC surges where you sort of get this more, kind of subcortical type, and then versus the more global arousal.
Mm-hmm.
Could you think of them as the same thing that's just on a spectrum? You have these ones that just kind of never get there. Or would you imagine that they're two really sort of distinct phenomena that actually are quite function distinct and have these different kind of reasons for occurring?
Yeah, I mean, you fully get it. It's super important question, and also, I mean, why, what are the mechanisms that make slow waves or low frequency activity become increased at the cortical level, although subcortically we have an activation, or we have a disappearance of spindles.
Yeah.
How that mechanism is put into place requires an understanding what happens to the different cortical layers in terms of how they are targeted by thalamic inputs, when there is spindles, when there is no spindles, and what the role of LC activity is cortically. And for the moment, I cannot tell you. I mean, we look at these LC searches, we identify them, and we can quantify their mean levels, but they are super, different in terms of there's multiple peak ones, there is single peak ones, and we don't know what features really make them different.
Yeah.
For the moment, I can say that it could be a graduation, so that maybe when LC activity does not reach a certain level, it might not be able to recruit other areas, like for example basal forebrain, cholinergic neurons. They seem to have quite a high threshold to be activated by noradrenergic input. So maybe that level would need to be reached, so that then you have the cholinergic input that activates the cortex. But it can also be purely related to how LC activity modulates thalamocortical circuits. Like that, this is a bit what Francesca Ciclari proposes. But yeah, we just need to test this. Like we need to, for example, inhibit basal forebrain
Yeah.
and see what happens to microarousals, or go into intralaminar thalamic nuclei and that are very relevant for microarousals, and see how modulating their activity impacts on microarousals, and what the role of LC fibers in that area is. So this is all sort of, we're doing the preparatory work for this. We are trying to inhibit LC fibers using novel tools. And I mean, it's all a huge amount of steps to be
Yeah, it's a lot of
Yes,
very
yeah,
careful experimental work.
yeah,
Joanne today it reminded me of something that a big interest for this group is the thalamus and thinking about the core and matrix thalamus and something that Bella has been thinking about with her sleep work. And I guess I just wondered if it's something that you've thought much about, but in terms of kind of, I guess, spindle generation and propagation, and perhaps whether they're local or global, do you think much in terms of, I guess, core and matrix thalamus in terms of this type of, I guess, yeah, how the spindles propagate, which cortical layers they end up in? Is that, yeah.
yeah. Oh my God, yes. That's what sort of I wanted to do
Yeah.
in my research
Yeah.
before we came on. We stumbled across the infraslow fluctuations.
Okay.
Um, So, I mean, what you have in the mouse is you have these very strong thalamocortical circuits in S1, where we really know the spindles that are generated at the level of S1. We can manipulate VPM, and we know that there is this causal relationship. We had done a study on this before. So we have really thought about mostly the core
Yeah.
ones, so the very simple spindles. But in the sense that they are generated in one thalamic nucleus and their manifestations in S1 deep cortical layers is very easy to make. But other types of spindles are probably generated in much more complicated circuits that involve different thalamic nuclei and also several ones.
Yeah.
So we have not really gone about these a lot.
Yeah.
That's a huge amount of circuit work that would need to be done
Yeah.
to
That's
clarify these.
interesting. Yeah. Wow. Um, Look, maybe Max has a question. You
Oh,
switched.
sorry, can I ask a
Yeah,
quick?
go
That
ahead.
was really,
Go
sorry,
ahead.
I missed the first part, and it was just lovely to see the ideas laid out. So in some of David's original work in Slice, he kind of showed this really interesting effect where noradrenaline had a particular impact on the thalamus and that it would depolarize thalamic kind of relay nuclei as well as the
Mhm.
RTN. But then acetylcholine had a differential effect on the relay nuclei and the RTN. I was wondering if you'd looked at any of that or tried to replicate some of those with more modern techniques, because I think this is such a fascinating sort of
Yeah,
dissociation in the circuit.
yeah, yeah. No, we have really looked mostly at noradrenergic mechanisms so far, but that's of course another very interesting question. Yeah. No, we haven't done that. But so my idea originally to look at LC was actually driven by the work that I had done in David McCormick's lab, where I had seen that in the slice, where at the time we had no optogenetics, no nothing, we could just record these spontaneous spindle patterns. And we were asking, why do spindles come and go? Why would they just disappear and not continue? And then when we put H-channel blockers, now we made spindles continuous. And then I studied what makes HCN, which is the hyperpolarization activated cation channel, why this one would just get activated out of nothing. Just within the spindling circuit with no noradrenaline, no nothing being liberated in an evoked manner. And what we found was that the spindles themselves, they lead to cyclic AMP synthesis most likely because the spindles themselves, they lead to calcium entry in thalamocortical neurons because they are generating calcium, low threshold calcium spikes. And that calcium activates an adenylyl cyclase. And that adenylyl cyclase then activates HCN channels. And the HCN channels then are a bit more active, and this depolarizes thalamic neurons. And this depolarization, it was nothing. I mean, it was 2 millivolts at the single cell level. It was super tiny, but it's apparently sufficient to drive an entire population of neurons out of the capacity to generate oscillations together with the TRN. It's a very, very sensitive membrane potential range that allows for spindles to be generated. And the cyclic AMP, I mean, it asks for beta-adrenergic mechanisms to be looked at. And so when I saw this infraslow pattern, it was clear that I first wanted to check LC and noradrenergic antagonists. When it comes to acetylcholine, then it would involve more also, muscarinic receptors that signal through, phospholipase C mechanisms that are less well characterized in terms of ionic mechanisms and what they really mean for spindles.
Yeah, I mean, I think you're totally right that this is a really complicated space. Please let me know if you guys go down this route because I think there's some really interesting implications for the kinds of information processing that could happen in downstream circuits and the
Yeah.
kind of role the ripples and spindles could play in that. It would be really fun to think about.
Okay.
I really,
Okay.
really love the work you're doing. So you feel free
Yeah.
to keep on with just the LC because it's equally fascinating.
Yeah, so I mean, when we did the pharmacology, we didn't want to start on any of all this huge mouse work and getting the mouse lines and so without having the pharmacology. So we started with that, and that turned out really that worked quite nice, no? I mean, it was challenging to put these drugs, but we got very nice effects on the inflow. So then it was clear should be an important driver. But it's clearly not the only one. And also, I mean, why the LC would generate these oscillations, it's probably a network phenomenon. It's not the LC on its own doing this. And you can then also ask whether, the feedback from thalamocortical oscillating circuits actually is the one that drives the LC because cortical circuits are very strongly innervating LC, no? So these are all open, open questions.
Bella, do you have a question?
Hello. Oh, thank you so, so much for your amazing talk. It was beautiful, and I find it all so fascinating. I guess
Thank you.
the questions are endless. Oh, no, thank you. The questions are endless. I guess one thing that you touched on was hippocampal activity that you want to look into more. Did you look at the coupling of these hippocampal shockwave ripples around these spindle events, or is that something you're still looking to do?
Yeah, no, so we have not gone into the spindle field into the ripple field. There is many people who are doing this in an excellent manner. So we have so far not done that.
Yeah, so
But
much to do.
yeah,
Yeah.
I really advise you to, have a look at the Asahara Oliva paper. I forgot the other author, but Asahara Oliva is, I think, one of the senior authors. And then there is also papers in eLife on, how exactly serotonin fluctuates in hippocampal
Yeah.
areas. It's also fluctuating on the infraslow level. And it shapes activity patterns in dentate gyros in a very marked manner. So really, it's really generating up and down states in dentate gyrus neurons. And that can, of course, then impact how activity is flowing in the in the hippocampus.
So
So
amazing. Thank you. I will look more into that.
have look serotonin and infraslow. There is one paper in eLife at least, and I think there is another one now. Yes. There is another one now from Yulong Li, which just came out
Okay.
on serotonin fluctuations. Yeah.
Thank you.
I feel
You're
like we could
welcome.
keep peppering you with endless questions. Does anyone else have anyone who hasn't said anything want a chance to ask something? Or I have one. I don't know if it's a small question, but maybe it can be the last one. So we stop taking your whole morning. But it seems like through all this kind of, history of looking at these different kind of the slow oscillation across species and, always roughly kind of in this 50-second time oscillation. I guess it somehow seems a little bit strange to me that we should see such consistency across species. If we imagine my autonomic set points and, the sort of range at which I fluctuate, I mean, it's probably a bit different to a mouse. I mean, I'm very short, so maybe I'm closer to a mouse than some people. But I guess, yeah, I just wondered, why we see such consistency in these really kind of hierarchical brain and body rhythms
Yeah.
across species that have such different ecological niches and such different sort of body
Yeah.
makeup.
Yeah. No, it's really yeah, it's very odd, no, that it should be so preserved.
Yeah,
On the other hand, we should think that, if we talk about 20 seconds or 50 seconds, it's already a factor of two, no? It's
yeah,
already between spindle and beta or low gamma.
Yeah,
It's the same difference.
yeah,
So I think it's just very difficult to come up with very precise measures because many of these, also the way we are measuring this, say with spindles or blood flow, these are all highly filtered signals that are also difficult to make phase relationships with. So, for example, the LC fluctuation, it's actually more 30 seconds. I think Natalie Hauglund or Julia Kirby in their paper, they have done a real spectral analysis. So it's actually slightly faster, but when you look at spindles, it's sort of how the network generates these patterns, and that's a low frequency filter, a low pass filtered signal of the LC activity. Yeah. So I think we have to be careful with all these frequencies. At the same time, I agree. Yeah. So there is now a study that maybe comes out soon, or it's already some were published, that in reptiles you have infraslow patterns of sleep-related activity that are reminiscent of, or at least Gilles Laurent says that they are more
yeah.
like reminiscent of alternations between non-REM and REM sleep, like as he says. So
Wow.
there is a huge debate on whether this is really REM sleep in these reptiles, in the squamates, no, whether this is an ancient version of our REM sleep. But it shows again that an alternation between two states on this tens of seconds timescale
Mm,
is preserved.
amazing.
I cannot really tell you why, but it could indeed relate to maybe some very ancient mechanism of how blood vessels are contracting, these very inert biological structures that take a very long time to do their job.
Yeah.
And that's just the pattern that's coming out in the measures that we take.
Yeah, I see. Yeah.
So yeah.
Fantastic.
Also in reptiles, you see blood fluctuations very strong on the
Mm,
infraslow time scale line. I think they
okay.
did that in chameleons.
Yeah, yeah.
So
Yeah, I remember the one from a few years ago, the science, I think it's a science or nature paper, but they're looking in the Australian dragon and they found the first kind of
yes,
the sharp wave ripple signatures in this lizard, which is very cool.
yes.
Yeah.
Yeah, so this is quite debated because not all
Okay.
people agree that this is really REM sleep. But
Okay.
maybe I can add here, so I didn't discuss this, but when we do a REM sleep restriction, so I didn't talk about the troughs at all. No, one of our studies is really on what happens when LC activity is low, and this activity must be low for REM sleep to be started. It
Mm,
cannot start otherwise. So we suppress immediately REM sleep as soon as we just activate a tiny bit the LC.
mm,
And so that gives this, that gives this idea that LC can actually consolidate sleep,
yeah.
non-REM sleep, because it prevents entries into REM sleep.
Yeah.
And it generates a spindle-free non-REM sleep state, but that is super, super stable.
Mm,
And that looks like it makes sleep better, if you just look at the architecture.
yeah.
Um, but so in a freely sleeping animal, we do a REM sleep restriction. So we make animals not enter into REM sleep. And we look what happens to the LC.
Mm,
Then we find that all of the animals desperately want to go into REM sleep.
mm.
They really want to fall in it as much as they can after a few hours of that restriction. They will always wait 50 seconds before they try again.
Wow.
You know, you,
Yeah.
you, they enter into REM sleep. We measure it and we interrupt REM sleep very mildly. So they wake up. So LC activity is high. Very briefly, they go back into non-REM sleep and they wait for 50 seconds before they try again. So this is an obligatory interval almost. You could say that they have to wait before they can enter into REM sleep. And it almost becomes a periodicity of 50 seconds of the ultradyne cycle. So you get non-REM and REM alternating a bit like in the reptile.
Yeah. 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 Oh, goodness. Wow.
So it's some mechanism that needs to complete in non-REM sleep
Yeah.
before you can enter into another state.
Nice. Fascinating. I keep promising that we'll finish, and then I keep asking questions. So we should actually
Yeah,
finish.
my answers are maybe a bit long, but yeah,
No,
we'll
no, it was
stop.
so fascinating. And to see, honestly, to see the whole story was so fascinating for me, and probably especially fascinating for students to, really see this scientific trajectory build up over years and so many careful investigations. It was really beautiful. Thank you so much. And thank you for being so generous with your time and spending so long with us.
No, no problem. It was really an honor, and I really appreciate.
Yeah, and
Thanks a lot.
we would love to keep in touch with you because I think everybody here has a lot of shared interests. So hopefully we will talk again, soon.
Absolutely. So, any questions of if you want me to clarify things, don't hesitate. And also any suggestions or questions are very welcome. It's just that, I think that it's going to require many more people to work on this because
Yeah.
there is that much that you can do also in terms of, mouse lines and it easily starts exploding. So I think it's going to be something that is going to be interesting for
Yeah,
many young scientists also to start
exactly.
thinking about.
Yeah. Thank you again so much. It was such a pleasure to have you, and we really appreciate your time. Thank you so much. Bye.
Thanks a
Bye.
lot.
Thank you.
Thank you so much for listening. Have a
Thank
good
you.
day.
You too.
Thank you,
Thank
Claire.
you.
All the
Thank you.
best. Bye bye.
Bye.